Right, yes, my name is Stephen. I'm from Melbourne University, and I'm going to tell you a bit about the work that we're doing in my group on genetic biocontrol. And here we go. And our group is called Piper Lab, which stands for Pipelines for Invasive Pest Eradication Research. And so we're interested in developing these tools for lots of different species. So the main technology that we're working on at the moment, which I'm sure most of you have heard of, is called GenDrive. And if you're not familiar with how GenDrive works, it's basically disrupts the normal way a gene is inherited. So normally you have a 50% chance of inheriting a particular allele of a gene or a copy of a gene. And so the frequency of that gene doesn't change in the population in the absence of any selection. But with a GenDrive inheritance, that distorts that normal probability of inheritance from ideally up to 100%, but more than 50%. So it basically increases in frequency in the population until you get 100% of the alleles in the population carrying the GenDrive. So if you target a GenDrive construct to a female fertility gene, then the outcome of that is it works as a suppression GenDrive that can result in eradicating or suppressing or even eradicating a pest population. And so this sort of technology has been quite well developed in mosquitoes. I expect it'll probably solve malaria in the next decade, I would think, but it's still got a bit of work to get the mechanism working properly for vertebrates. So the success of developing it for vertebrates will depend on not only an efficient GenDrive design to get that mechanism working efficiently, but also simply the logistics and the technicalities of actually just producing animals that carry GenDrives, these non-laboratory model animal species that we need to target. So for a good GenDrive design, we need suitable target genes for female fertility and development. There are already enough known, well-characterised candidate genes that we can target for that. For example, genes that produce the zona pellucida that Ellen just mentioned in the previous talk. And we also need efficient copying of the GenDrive in the sperm cells, the spermatocytes that form sperm. So that's where the actual copying of the GenDrive actually occurs, or needs to occur, because the reason for that is that it has to exploit this particular phase of meiosis, which is when you get this crossing over between the maternal and paternal chromosomes, and it exploits that mechanism to increase the efficiency of the copying of that GenDrive. So to sort of go over it very quickly how that works in a spermatocyte. So it exploits, or an engineered GenDrive exploits the CRISPR-Cas9 tool that was developed a few years ago, and it actually incorporates a gene for Cas9 in the actual GenDrive construct that's driven by a promoter that switches on that gene in the spermatocytes, and that gets targeted to a female fertility gene, and it also carries within that cassette a guide RNA, and that's basically allows the Cas9 enzyme that causes a double-stranded break in the DNA to sort of target to a particular site in the genome, which in this case is the female fertility gene. So the Cas9 cuts the site, and then the natural mechanism in the cells is then to use the GenDrive version of that allele to sort of repair that cut version of the allele, of the wild-type allele, and that essentially creates two copies of the gene. So that's how it propagates through the population. So then that means that all of the offspring ideally will inherit that GenDrive. So that's so basically in my lab I have a student, PhD student, Clancy Lawlers, whose project is basically to optimise the efficiency of this copying of the GenDrives, and this is using the zebrafish as our sort of standard vertebrate laboratory model, because it's a very efficient system to work with. And he's doing a proof of principle GenDrive, where he actually sort of separates the different components for sort of biosecurity reasons into different parts of the genome. So he's targeting the CYP19A1A gene, which is basically essential for producing estrogen. And in the context of a zebrafish, that causes sex reversal. So it basically causes animals that would normally develop as females into males. So he's engineered these fish already. So you can see here these this red fluorescence that you're seeing in this fish larva here indicates the insertion of this partial GenDrive module. And then there's another component that he's also inserting elsewhere in the genome, and then by crossing these fish, he'll be able to basically demonstrate the actual, what will hopefully be the first functioning GenDrive in fish or invertebrates. And so that's one part of the research that we're doing in the lab, is to produce improve the efficiency of this Cas9 dependent GenDrive, and the other side is simply to get these GenDrives into non-model species. So this is basically something that's easy to do in laboratory mice, but using those same techniques in mice is not really applicable to other species. So the species that we have, these are just ones that we have funding for in the lab. So I have federal government funding for carp and cane toad, and also philanthropic funding for fox. This is just this guy who really doesn't like foxes, and also a small amount that I initially got from the Rabbit Free Australia Foundation. That was, although it's a small amount, it really helped to sort of kickstart this research in my lab. There's notable omission, of course. There's no funding for feral cats currently. For this research. So the strategy or the pipeline for carp and toads basically depends on injecting the CRISPR components into eggs. And the advantage of this is you can just inject into thousands and thousands of eggs, because doing a targeted knock-in is not necessarily a very efficient process, but you can do lots of them and then easily grow them up. For mammals, it's you don't really have that luxury, especially with non-model species. So there's a sort of a different strategy that we're using, which depends on, first of all, having cultured cell lines, and then using the technique called nuclear transfer. And those of you who are familiar with the cloning of Dolly the sheep a couple of decades ago, this is basically the technique that was used for that. So you can do your CRISPR editing in the cultured cells and then use those cells to produce embryos via nuclear transfer. So this is the reason for adopting this strategy. Is it's a strategy that could potentially, once it's developed and set up in our lab for one species, it's more easily sort of translatable to any other mammal species, as opposed to sort of the methods that are used in laboratory mice, which are not translatable to other species of mammals. And the scenario of how this works, for example, for the fox, how we're trying to develop for the fox, is first of all, we've established the cell lines from the fox already, and you can see on the right here these are the cells expressing a red fluorescent protein that indicate this process of converting them into a sort of stem cell state called iPS cells. And then the idea then is to be able to insert the GenDrive into those cells once the design has been optimised with the zebrafish work, and then to use oocytes, ideally from foxes, from hunted foxes, or potentially, or just from using oocytes from vet clinics, from DSX dogs, and then using those oocytes to nuclear transfer and create embryos and be able to cryopreserve them, and then wait for the next big pot of money to actually start producing captive foxes to do all that. So there's a few caveats with all of this kind of research. For one of them that's often pointed out is the risk of introduction to non-target population, a similar thing that Ellen just alluded to previously. So, for example, if cane toad were to get back to South America, where they come from, either accidentally, which is unlikely, or maliciously, it's actually easier to engineer a GenDrive resistant allele than it is to actually engineer the GenDrive in the first place. So although it's a very unlikely scenario for this to happen, we do have that safety backup option to protect any non-target population. So obviously, I haven't really talked about cats very much, but it's got really the opportunity to come here is sort of, I've been thinking for a long time about feral cats, of course. In the case of cats, I mean, they do have a longer generation time compared to other animals, and it takes longer to breed them up in captivity. So this would be many decades of to actually sort of have an impact on the population once it's deployed in the wild. But this would require sort of constant monitoring and probably breeding up very, very large numbers of GenDrive animals in captivity for release. So it would have to be a very long, sort of large, continuous investment into that deployment. And of course, we're going to have an inevitable resistance from crazy cat owners, even if they're a minority. So there's this, that aspect to deal with as well. And in terms of how that might be dealt with, I mean, GenDrive is, for most of the invasive species, we don't have this issue with domestic cat owners or domestic owners of the species that, a lot of people will be resistant about the potential for the GenDrives to get into cats. But I would envisage that in the future we might have some kind of a legislation where we actually mandate domestic cats have to have GenDrives and have to be bred by licensed breeders, some sort of a regulatory framework like that. I envisage probably what we would see in, say, 50 years' time or hopefully less. Thanks for your attention. I'll leave it there.